Humans have noticed for a while that children show some of the traits of their parents, but they've also noticed that they don't look exactly the same. So we do understand that there is some sort of passing of traits, but the true method of how it happens has really not been understood until this past century when we've looked at what we call genetics. So the father of genetics was a monk named Gregor Mendel, and he worked with pea plants. There are reasons why he chose pea plants. First, they were hybrids of them could easily be produced, so you could take one strain and combine it with another one. There are also lots of different varieties of pea plants, and they were small and they were easy to grow, and maybe most importantly, they could be fertilized. One plant could fertilize itself, and that'll come up later when we talk about some of the experiments he did. Before we get started, though, we need to know some of the terms of breeding or of genetics. One is true breeding, and this is where you take a plant, and let's say you're looking at some trait, let's say it's purple flowers, and this will come up again, and you self-fertilize it. So you take that one plant and you can just make it mate with itself. Mate's not the right word, but fertilize. And then all of its progeny will have the same trait. So if it has purple flowers, you have it self-fertilize, it will make seeds, you plant those seeds, those grow up and they'll have purple flowers as well. And if you had white flowers, and you self-fertilize those flowers, it would also show those same white traits, white flowers in their progeny. Okay, so some other ones. A monohybrid cross is where you take two true breeding plants that are different. So you have purple ones that true breed and white ones that true breed, and you cross them, and they only show one trait. So all of the progeny will have purple flowers in this case. Those progeny that they have, all those purple flowers, would be called the F1 generation. So the offspring produced by crossing two true breeding strains that have a different trait. The F2 generation would be the self-fertilization then of the F1 generation. Okay. This figure here shows the process of or of fertilization from one to the other. Take white pollen and you can fertilize a purple plant, and you can do the same thing in the other direction. So what they found here, this is a monohybrid cross. They took purple and white true breeding plants, and all of the progeny, all of the plants that came from that, had purple plants. The trait that is expressed is called the dominant trait, while the other trait that's not expressed is called the recessive trait. And what Mendel found was that no plants had an intermediate trait, so none of them were pink or light purple or anything like that. They were either purple or they were white. And this disproved a inheritance pattern or theory at the time called blended inheritance, where that all the traits were just kind of two parents were just blended together, and children were a blending of their parents. So, some of the things Mendel did is he took the F2 generation, which again was the self-fertilization from the F1 plants, and he realized that in the F2 generation that recessive trait showed up again. And it was always in this 3-to-1 ratio. But when he self-fertilized those, so making an F3 generation, he found that some of them had all dominant trait, and then some of them had actually some of the white trait, some of the recessive trait, and then true breeding, the white ones, they always had the recessive. So what was originally thought as a 3-to-1 was actually a 1-to-2-to-1 ratio. 3/4 of them had the dominant trait, the purple, and 1/4 of them had the recessive trait, which was white. But then that self-fertilization of those found that one of those purple ones was a true breeding dominant plant, two of them were not true breeding, and one of them was true breeding recessive. So the first principles that Mendel came up with was called 5-element model. The first one is that parents transmit discrete factors, and we're going to call them genes. The word genes hadn't been coined yet, so he just called them discrete factors. But we're going to call it genes from now on. Each individual receives one copy of gene from each parent, but not all of the genes are identical. So an allele is an alternative form of the gene. So here in our example, we had purple allele and we had white allele. If a P has two of the same allele, so if they have two purple alleles, then they are homozygous. If they have one purple and one white allele, then it's called heterozygous. Also, the alleles remain discrete, so there was no blending. You couldn't change a purple into a pink or a pink into a white. They were discrete. And just because an allele was present didn't mean that it was going to be expressed. So the dominant allele was always expressed even when it was paired with a recessive allele, and the recessive allele was only expressed when it had two of them. So using these genes, we can then determine what a genotype and phenotype are of individuals. A genotype would be the total set of alleles of an individual, and the phenotype would be the physical expression, either purple or white, which led to Mendel's first principle with the principle of segregation. So this is talking about alleles that are on homologous chromosomes. So two alleles per gene segregate or separate during gamete formation, and then at random are rejoined during fertilization. And this has to do with the way the chromosomes separate during meiosis. Okay. Mendel didn't have any knowledge of meiosis or chromosomes for that matter. He just knew things in terms of traits. And he, through his experiments, realized that they separate individually. So, one of the things he came up with is Punnett squares, where you are tracking these traits and looking at different ways or probabilities of different ways that they are going to combine. So when you cross a purple flower plant with a white flowered plant, you're going to use letters to denote the dominant and recessive alleles. So a big P is purple, and a little p is white, and a true breeding white flower is little p. It has both recessive, homozygous recessive. A true breeding purple plant is big P, and then a heterozygous would be big P, little p. Alright, so real quick, I'm going to just show you what this would look like. Okay, so here I have a purple plant, and we're going to say this is a true breeding purple plant. So it is big P, big P. Okay, and we are going to cross it with this yellow plant, which we're going to call little p, little p. Since it's recessive and it is yellow, it has to have the same. And we're going to predict what would happen if we fertilize these two plants. And so to do that, we're going to have one going across the top and one going across the side. And what we're going to do is put the alleles on the top. So I'm going to take this P and I'm going to write it up here. This is going to be a capital P. And we're going to take this other P and we're going to write it over here. Capital P. And then we're going to do the same thing over here. This little p is going to go right here. This other little p is going to go right here. And then to determine what the ratio of their progeny are going to look like, you would just carry them down and put them into the box. So this little p, big P, is going to go into this box. And this little p is going to go into this box. Then we're going to do the same thing for the next one. Big P here. Little p there. And the same thing over here. I'm not going to draw the arrows. Big P, little p, big P, little p. Okay, because big P is dominant to the little p, all of these will have purple flowers. This one will be purple, this one will be purple, this will be purple, and that one will be purple. Right? That's how you do a Punnett square, and that's how you would determine what the phenotype or what the expression of the gene is going to be. Alright, so now let's back up. Let's do one more just for fun. Let's say that this one is actually a heterozygous. So even though it's purple, it has one recessive allele. And then this one's still yellow, so it has this recessive allele. Okay, to do the Punnett square, if we fertilize these, what are their progeny going to look like? We're going to put the big P over here. We're going to put the little p over here. And just like before, put a little p and a little p. And then we'll carry this down. Big P here. Bring that over. Little p there. Big P. Little p. Okay, so what color are these going to be? Well, they're big P, little p, so they have the dominant allele, so they're going to be purple. Okay, this one is going to be little p, little p, little p there, and so is this one. So these, then, because they have two recessive alleles, are going to be yellow. So we would expect that if we crossed these, that the progeny would be half purple and half yellow. Okay, and that's how you do a Punnett square. That's how you conduct these probabilities. We're going to do a lot more of these on the worksheet, though. Let's back this up. Okay, that's how you do a Punnett square. Here are some dominant and recessive traits, examples of them in humans. We've talked about, let's see, sickle cell anemia in class, which is a recessive trait. We talked about Huntington disease, which is a dominant trait. If you have that, you're more than likely going to pass that on to your children. But there's some other ones here. You don't have to know all these. These are just examples. Okay? So, again, just to reiterate, a dominant trait only requires one copy of that gene for expression, whereas a recessive trait requires two. These are pedigrees showing a dominant trait. So if it's colored in, it has this dominant trait. and because it only needs one copy for expression, the progeny are more likely to have it expressed. Whereas a recessive trait, you're going to see a lot less expression. Alright, a dihybrid cross then gets a little more complicated because here we're looking at two separate traits in a single cross. So we're looking at, in this example, the color and then the texture. So it can be either yellow or green, or it can be either smooth or wrinkled. Okay, and if you have two true breeding plants, meaning it's homozygous dominant and homozygous recessive, and you cross those, all of the progeny are going to have the dominant trait, brown and yellow. Okay? If you allow them to self-fertilize to produce the F2, okay, you have the dominant trait, but all of them are heterozygous, meaning they have the recessive traits just not being expressed. The F2 will show this ratio of 9 to 3 to 3 to 1. So 9 of them will be round yellow, have both dominant traits. 3 of them will be round green. 3 of them will be wrinkled yellow. And then 1 of them will have both recessive traits, wrinkled green. Okay, that 9 to 3 to 3 to 1 ratio is an important ratio to remember. And we will do a dihybrid cross in class. So this led to Mendel's second principle, the principle of independent assortment, which is kind of like segregation, but it's talking about genes on different chromosomes. So in the dihybrid cross, the alleles of each gene assort independently. So just because something's green does not mean it's going to be yellow. Or sorry, just because something's green does not mean it's going to be wrinkled or green smooth. That those traits are independent of each other. They show random combination when they come back together in fertilization. So to determine probabilities, what we do is we add up, we use two different rules. The first one is rule of addition, where you add the independent events, the probability of independent events, and the sum is the probability of those two events occurring at the same time. So when you're crossing a heterozygous purple plant, the probability of producing a heterozygous is, if you count up the Punnett squares, okay, here's our two heterozygotes, same thing here, same thing. And you want to know the probability of having a heterozygous, well then you just have to add, this is 1 out of 4 plus 1 out of 4. It's going to equal 1 out of 2. So you have a 50% chance of having a heterozygous. It's pretty simple. Multiplication is the probability of two independent events occurring at the same time. It's going to be the product or the multiplication of their probabilities. So this is, so the probability, an example of this is if you have males and females. Females have two X chromosomes. Males have an X and a Y. If a male and a female are producing offspring, what is the probability that you will have an XY offspring? Well, females can only get an X, so the probability of obtaining an X from the mother is 1. And the probability of obtaining a Y from the father is 1/2. It can either be X or Y, 1 out of 2. And then you multiply those together, 1 times 1/2 equals 1/2. That's the probability of getting a male, which is, that's what this map is. Shows the proportion of countries that have more males and the proportion of countries that have more females, but it's generally worldwide about 50%. Alright, so one thing we can do to determine if a phenotype is X or Y, sorry, getting my terminology mixed up. So one thing we can do to determine what the genotype of a, let's say, purple flower is to do a test cross, and that is to cross it with a homozygous recessive. Okay, and based on the ratios, you will know what type of plant you have. So again, we're going to use our little Punnett square. Okay, and let's just say we don't know what this purple plant is. We know that it has one big P here, but the second allele we just don't know. P question mark, or this is often abbreviated as P underscore, so there's the blank there. So we're going to do a test cross. Okay, if this is a little p and this is a little p, so we know every time that we cross this with a yellow flowered pea plant, it's going to have a little p. If our two options then for this pea plant are, it can either be big P, or big P, little p. And so let's say that it is big P, big P. What are the flowers of the progeny going to be? Well, they are all going to be big P, little p, right? So if I ask the question, what is the genotype if all of the flowers of this cross are purple, you would know it's big P, big P. 100% purple. But what if it's big P, little p? And we already did this test cross before, but all of the progeny would not be the same in this case. In this case, you would have some of them, which would be purple. In fact, half of them would be purple, and half of them would be yellow. So again, if we don't know what the genotype is, but we do know the phenotype, that it's purple, we can cross it with a homozygous recessive, and they will show either, if they show any yellow, then we know it's big P, little p. If it shows all purple, then we know it's big P, big P. Okay, that is a test cross. Now, the principles that Mendel did come up with were very good, but they don't cover all traits. We have his assumptions included that the traits that he looked at, they were controlled by a single gene. They only had two alleles, and there was a clear dominant recessive relationship between those alleles. But most genes don't meet these criteria, as you can tell. So these are my parents. These are all my brothers and sisters. Yes, I have a big family. But if you just look at their hair color, they both had black hair. We have some of my siblings that have very black hair, but most of us have brown hair. And then we have one of my sisters has red hair, another sister has blonde. So they didn't follow these same rules. So there's some other things going on, including one of them, which is called polygenic inheritance, where you have multiple genes involved in controlling of one trait. These traits show continuous variation, or what's also called a normal curve. And an example of that is height in humans. We aren't either tall or short. We can be a variation between the two. And you look at the heights of my brothers and sisters, we have variation in our height as well. Pleiotropic is another effect where one gene controls multiple different expressions. Usually one of the traits is known, and then the other things are unknown or are shown after the mutation to that allele. So an example of that is cystic fibrosis or sickle cell anemia. Only one gene controls multiple different expressions. So that's what this figure is. These are all the expressions of cystic fibrosis, which is very large, but is really only controlled by one gene. Some traits can be controlled by more than 2 alleles. So, for example, blood types in humans have 3 alleles. And that's what this is down here. The actual allele expression is a little complicated, but basically you have more than 2 alleles showing the expression of blood type. So the number of alleles for any gene to express is constrained, but within a population there are more than 2 being expressed. Another one is called incomplete dominant. So this is where the combining of 2 alleles shows an intermediate between the two. So if you had red or white and pink flowers, were the result of a cross-fertilization. And then the last one is codominance, where both are expressed. So this happens again with blood types, where if you have an A allele and a B allele, then you have type AB blood. Environmental influence is also where the external environment shows expression of traits. So this is an example of this is found in Siamese cats. All of the genes are the same on the cat, but the ones showing color are affected by the temperature. If the temperature is below 33 degrees, then it will be expressed as black. It will release pigment. If it's above 33 degrees, which is found on the core, the center of the cats, then it will not show pigment and the fur will be white. Epistasis is another phenomenon where 2 genes have to be expressed in order for a trait to be expressed. So Emerson did a test cross of 2 different varieties of corn, and what he found was that they had to have both the A allele and the B allele, the dominant form, in order for them to be purple. If they had the dominant A allele but not the dominant B allele, or vice versa, then they turned out to be white. And so this showed a different ratio. This was a 9-to-7 ratio, which was different than the 9-to-3 to 3-to-1, which he had expected. Alright, that's it for our first chapter of genes. We're going to do more genes in a separate lecture.